Hello students, in previous video lecture we already discussed about the basic concepts of eukaryotic transcription and the steps that are required to carry out the process of eukaryotic transcription. Also the factors required for the process of initiation we also discussed in the previous video. Now we will going to discuss about initiation of eukaryotic transcription in details. So process of eukaryotic transcription includes, there are three sequential steps. First is initiation, elongation, termination. We will discuss about the initiation. We already know about the initiation factors that required to carry out the process for transcription of eukaryotes. Transcription initiation that describes the assembly of pre-initiation complex and promoter clearance. Transcription in eukaryotes starts with the binding of TBP, that is Tata box binding protein, to Tata box, and then recruitment of transcription factor 2B. After that there is a recruitment of RNA polymerase 2 and transcription factor 2F complex, and this will stabilize by the factor that is transcription factor 2A, and after that there is a binding of transcription factor 2E. The RNA polymerase, when bound to the transcription factor 2E, that recruits the transcription factor 2H, and the main lead role of this process is done by the transcription factor 2H. It generally open up the DNA and phosphorylate the CTD of RNA polymerase 2, mainly the RNA binding protein 1. So complex of transcriptional factor and RNA polymerase 2 assembles at core promoter, and this will called as pre-initiation complex. Here we can see that a pre-initiation complex in which Tata binding protein is first bound to the promoter site. After that transcription factor 2B, then it recruits RNA polymerase 2 with the help of transcription factor 2F. Transcription factor 2F mainly prevents the non-specific binding of RNA polymerase 2. Then transcription factor 2E, it recruits the transcription factor 2H. Transcription factor 2H that performs the main function, that is separation of DNA, and it catalyzes the phosphorylation of CTD. After the formation of pre-initiation complex, the next step is formation of open promoter complex. Once all the transcription factors and the RNA polymerase 2 assemble at the co-promoter, that is, this is your Tata box is the co-promoter. When all the transcription factor and RNA polymerase 2 that assembles at the co-promoter, transcription factor 2H, this is the hero of the transcription initiation process in eukaryote, that catalyzes the ATP dependent separation of DNA strands at initiator sequence, that is INR region. So transcription factor 2H, it requires the help of, or it is dependent on the ATP that gives the energy to separate the DNA strands at initiator region. This will lead to resulting the exposure of template, that is we required DNA template to carry out the process of transcription. This will lead to resulting the exposure of template, and that convert closed complex into the open complex, and this once will lead upon the complex form. Transcription factor 2H phosphorylate the multiple serine fifth residue in CTD, that is carboxy terminal domain. In this note is phosphorylated RNA polymerase 2 initiate the polymerization of ribonucleotides in RNA transcript and undergoes the process of promoter clearance. That is phosphorylate होने के बाद RNA polymerase 2 का काम क्या है कि अभी राइबोन्यूक्लियोटाइड्स ऐड करना है, मतलब पॉलीमराइजेशन करना है, तो वो करने लगेगा, और प्रोमोटर क्लियरेंस मतलब प्रोमोटर जहाँ पे वो यहाँ पे बाइंड हुआ था अभी वो साइट वो छोड़ने लगेगा और आगे मूव होने लगेगा. ठीक है. So this is all about the transcription initiation process in eukaryotes. In the next lecture, we will discuss about the transcription elongation process in details. Till then, if you have any question, you can ask in the comment section. Thank you.